Just to clarify, these are two talks that are listed in the program as CAFA Predictor 1 and CAFA Predictor 2, and that we left blank because we didn't know who would give the talks. So the first talk is by Florian Boecker from Crop Bioinformatics at Bonn University. And the second talk will be from Shangfeng Zhu, who we heard so much about, from Fudan University, right? Okay. Florian.
Yeah, thanks for the introduction. First of all, I think heads up to the ZooLab guys. Unfortunately, we weren't nearly as often in the top 10 as them. So I'm from the group Crop Bioinformatics from Professor Schof at the INRES Institute at the University in Bonn. And I want to talk about our tool AHRD that uses lexical description for the CAFA 3 challenge. So AHRD focuses on human readable descriptions. We assign concise and trustworthy human readable descriptions to proteins by emulating the decisions made by human creators by performing a lexical analysis on candidate descriptions from sequence similarity search results. And we also transfer geo annotations with those descriptions. AHRD is written in Java and built by AHRD, and you can download it from our GitHub repository at github.com/groupschof/AHRD. So a scientist's first contact with the function of a protein is often through many possible descriptions, for example, in a BLAST search. But then the scientist has to weigh a few different options against each other. The naive approach is to take the BLAST result with the highest score. But of course, we do also have BLAST results from multiple different databases. In this example, Swissprot and TER being curated. But then on the other hand, Tremble not being curated. And you already see we have descriptions in here like putative that start with putative and stuff like that. So these descriptions probably come from computational methods, and we have to deal with them. So human readable description should obviously give a hint at the function, be short and be correct. But human readable descriptions, especially non-curated databases, are often transferred from homologs, contain sometimes very specific information. For example, the clone ID from the specific experiment are automatically assigned and thus then contain words like similar to or hypothetical, or sometimes are just wrong. So for this reason, the first step in AHRD is to filter all descriptions through a blacklist. This blacklist looks at the start of the line and recognizes words like putative or similar to remove protein descriptions that have been transferred computationally from other proteins. And then sometimes you even have proteins in there with descriptions that aren't descriptions at all, like uncharacterized protein, and we remove those from the pipeline completely. Then at the next step, we assign a score to each word in each description. If the alignment score and the overlap between the query sequence and the target sequences is good, that increases the score. Otherwise, the score is decreased. If the frequency of the word in the BLAST hits is high, the score goes up. Otherwise, it goes down. And if the description comes from a curated database, this increases the score as well. And if it's from a predicted database, like Tremble, we decrease the score. We then assign each description a score based on the words contained in it, the frequency of the combinations of those words, and the alignment score. So here in this example, AHRD chose a zinc knuckle protein, and at the bottom you can see the accompanying geo annotations. And if we compare that with the best BLAST method, it used ATP-dependent RNA helicase in Swissprot, in TER, the best blast hit would be cold shock domain, and in Tremble it would be the Vasa-like protein. Blast2Go, just to show an example, also chose the best blast hit from TER. So why did AHRD choose zinc knuckle protein? Well, if you look at the, first of all, we have a zinc knuckle protein in a highly curated database, so in TER. But then you also have this mention of zinc finger and zinc knuckle in a few descriptions in Tremble, which is the reason AHRD chose it. And through an amazing coincidence, it actually had this description in the actual reference in Uniprot KB. So it was a zinc knuckle protein. And at the bottom at the left, you can see the geo-annotations from the database versus our prediction. So you see the zinc ion binding term was predicted accurately. Then, of course, there are some other terms that are totally different. But then you also have this nucleic acid binding term on the left and the DNA binding term on the right. So we have this problem again, which is very close to the example Ido just showed, that those terms are almost the same, but if you use the naive approach of looking just at the cardinality of terms that are in the sets or in the shared set and stuff, then you're not getting the whole picture. For that reason, we are using semantic similarity to evaluate our Go annotations. We do so by counting all whispered annotations in all three ontologies, and we propagate those annotations up the tree. We then calculate the annotation frequency by dividing each count for each term with the count of the respective root term. This results in an annotation frequency for the root term of 1 and for a very specific term with a very low value. We calculate then the information content by using the negative logarithm of the annotation frequency, which results in an information content of 0 for the root terms and value of between 10 and 20 for very specific terms that have been annotated not very often. You then can calculate the maximum common information content of two terms by looking at the highest information content in their shared ancestry. And from that, you can calculate the semantic similarity of sets of GO terms by summing up the maximum common information content between those two sets. So here is an example. We have a ground truth term here and a prediction, which is very close. And we then can calculate a recall by dividing the maximum common information content of those two terms with the information content of the ground truth, and a prediction by dividing the maximum common information content of the two terms by the information content of the prediction. So if you want to do that with sets of terms, it gets a little bit more complicated. I won't go into the details, but we define it. We define the recall as the fraction of the ground truth's information content that the prediction was able to reproduce, and the precision, the fraction of the prediction's information content that was actually part of the ground truth. And then we calculate the F-score as the balanced harmonic mean of recall and precision. So AHRD also has a few parameters. We assign weights to how much the alignment score is used. But then also the question is how much does the overlap score between the query sequence and the target sequence, how much does that weight in? And of course also the confidence in the different databases. These parameters need to be trained. For that reason we use a genetic trainer, which is fairly simple. The first population is filled with random parameter sets, and then for each generation we evaluate all parameter sets, rank them according to their mean semantic similarity F-score, and generate the next population by letting the best 20% survive, generate 20% of mutants from the best 20% and 20% of recombinations of those best 20%, and then fill the rest of the next generation, the rest 40%, with new random sets. This guarantees that the score of the best parameter set always goes up, and works fairly well because you quickly get an enrichment of really good parameter sets in those top 20%. Of course, if you train, you also need a reference. You also need a ground truth. For that reason, we built our own test set. We used SwissProt, clustered it with the CD hit algorithm at a 20% sequence identity setting, and then removed all proteins without experimentally verified Go annotations. So we call this the XGOA version. After that, we removed all proteins with descriptions we deem undesirable. So we just used AHRD description line blacklist I talked about before. As a result, we have 2,251 proteins remaining with over 74,000 GO annotations. So to train AHRD for CAFA 3, we created a SwissProt version without the test set. So our scores are not inflated by self-matches. We, of course, then perform blast searches on the special version of SwissProt and on Tremble, and then randomly split our protein set 10 times in 1,251 proteins for training and the remaining 1,000 proteins for evaluation. Then we trained and evaluated AHRD on each of those splits by performing 100 generations with 200 parameter sets per generation. And then we just used the parameter set that had the highest evaluation score at the end. So, of course, we also submitted 3 different methods. Our first method, we call it AllGoa, where we just used AHRD on all CAFA 3 targets, searched in the normal SwissProt and Tremble database. Our second method we call XGOA, where we tried to up the precision by trading for recall by creating first a version of SwissProt that only has and Tremble with only proteins that have experimentally verified Go annotations, because we think the quality of those annotations is better, and used AHRD on those targets, on the CAFA 3 targets in those databases. The third method is basically the opposite of the second method, where we said, okay, it's okay if precision goes down, but we want to have a high recall. And we knew that CAFA 3 focuses on new annotations. So we thought why not just remove all 100% hits from the blast searches? So AHRD is forced to take the second best and thus get new annotations, because we also knew that the CAFA targets came from the UniProt databases. After using AHRD on these sequence similarity search results, we then merged them with the predictions from method 2. So basically get both of both worlds, with the trade-off that the precision will probably go down. And in the end of June, I think, you released 1,300 and something benchmark proteins. And I thought everybody is going to present their results respective to those benchmark proteins, but nobody did. So here are mine. And they're not good, as you see. The precision, the recall is kind of meh, but the precision is really poor. So why is that? And we looked at the benchmark annotations and saw that, for example, 142 proteins only had one annotation, namely the GO annotation for protein binding, and another 126 also had only one term, which stands for structural constituent of ribosome. We suspect this is based on high throughput experiments that only tested for those specific terms. And AHRD even correctly annotates 49% of the 126 proteins with the structural constituent of ribosome, but AHRD also predicts a lot of other terms, because, as you've seen before, the terms of a database entry are all transferred. So we found out that in general, the number of new annotations we got from CAFA 3 is very low compared to the number of annotations you have in UniProt typically, and also very low compared to the number of annotations we typically give. So what we thought is, why not just level the playing field a little bit and make up a ground truth that is not only based on the new annotations, but also on the annotations that were previously known. So we took the mapping of the target IDs to the UniProt IDs that was supplied with the targets, and merged the UniProt annotations with the CAFA 3 benchmark ground truth. And this increases our scores quite drastically. But of course, now the few new annotations are washed out by all the old ones. So this is, of course, to be taken with a huge grain of salt. So with this, I want to close and want to thank the National Science Foundation for making travel to this meeting for me possible. Also want to thank the CAFA 3 staff for organizing this cool challenge, my university, and Hassi Salab and Katrin Klee, who worked on AHRD before I did, and of course my professor, Heiko Stoff. So, thank you.
Any questions?
Thanks for the talk. Have you read our article on our prediction method called Panzer? It does this description prediction.
No, I didn't.
Okay, 2015
Yeah.
in bioinformatics.
Yeah. Okay, I will.
Yeah.
Okay, this is left to be talked about. If there's no other questions, let us close this session and... No, there's a break. 10-minute break.
Oh, 10-minute break. Yeah, just a 10-minute break to bio break to stretch our legs.
Yes, this is a short break, and then the Jurulab will present right after.